Hi everyone, it's Andrew, and today I am going to be doing a viral transduction. So first things first is you're going to need to prepare a solution of somewhere between like 10 to 20% bleach. So someone was actually already in here before me doing a viral transduction as well. So I already have the solution of bleach sitting in here. This should be enough for me. But let's prepare another one of these baths just for good measure. So usually there are a bunch of flasks over here, and don't need to be super exact here, just kind of eyeball like somewhere between 1 and 200 mils of bleach, and then I'll usually fill that up to around 500 mils with water. Put that in secondary containment, and then bring this over to the hood, and definitely need to spray this with ethanol around the sides before we bring it in. And then here, just be very careful. Of course, this is glass and it's slippery, so you don't want to drop it and break it. All right, now I'm also going to sterilize my work surface because someone was in here earlier. There are these plates here that are full of bleach, and you'll see why that is later on, that we leave the bleach to sit with the virus. So we're going to be harvesting lentivirus today and transducing Jurkat T cells with the lentivirus. So lentivirus is a type of retrovirus. So it uses a reverse transcriptase to integrate a RNA virus into the genome of cells. So because of this, it allows us to insert payloads that we're interested in into human cells of interest. But this also makes the virus really dangerous because, well, if it's infecting the human cells in our flask, then it can, of course, also infect cells in our own bodies. And so when you're working with lentivirus, you need to be extremely, extremely careful. You need to follow all the proper safety protocols, making sure you're wearing full PPE, lab coat and gloves, that you have these solutions of bleach ready at your disposal, just in case there's any spills. Always mop it up with bleach, not with ethanol. And yeah, I think just, you know, respect the lentivirus, be careful with it, and it's not so bad. So what I'm going to be doing here first is plating the Jurkat cells to prepare for them to receive the virus on top of them. So here I have this flask of Jurkats that I passed on Monday. I'm going to harvest these cells now and passage them at the same time into a new T75. So these Jurkat T cells are a type of suspension cell, which means that they don't adhere to the bottom of the flask. And this makes them super easy to passage. All we have to do is take some portion of these cells, put them into our new flask, and then put media on top of them. This is unlike adherent cells, which we would need to first add trypsin to the plate to actually lift the cells off of the bottom of the plate before we can passage them. And I'll probably make a video about how to passage suspension cells sometime in the future. So because we need a lot of cells today, I'm just going to harvest everything that is in this flask And then we are going to spin this puppy down and see how many cells we have in this tube. So I will spin these cells down at 400 G for 4 minutes, after which we will resuspend in fresh media and count. So we have about 20 mils of media in here. So I'm going to grab my 20 mil balance and hit go on the centrifuge. So I'm going to wait to make sure this is coming up to speed before walking away. Looking good. And now I'm going to very quickly calculate how many cells I need. So I'm plating cells for Matt today as well. He needs 27 wells. I need 25 wells. So in total that's 52 wells of jerkats. Basically 52, including untransduced, unique samples that we need to produce and test. And I'm going to be plating these cells at a concentration of 1 million or 1 e to the 6 per mil. And I'm going to eventually be plating 500 microliters of our cells at this concentration in each well. So in total we're going to need 26 million jerkats. So this is the number that we're hoping to hit off of the cells that I just started spinning down. So yeah, we'll count and then we'll see how it goes. In the meantime, I'm gonna grab a few 24 well plates. Looks like we'll need 3 in total. Matt and I can just share our untransduced wells on one of these plates. And I'm also gonna label my passage flask as well as bring in my jerkat media into the hood. So you'll notice that you know, I'm following sterile technique as I bring stuff into the hood, spray everything down with ethanol before I bring it in, except for these flasks. And the reason I don't spray these flasks is because you can see here there's a little like piece of filter paper that lives on this cap that allows the passage of gas to get in and out of the flask. And we don't want to saturate that filter or else it'll stop the passage of gas into our cells, and well, they won't like that because they won't be able to breathe. So that is why in this one very specific instance I do not use ethanol. So you'll notice on every flask used in tissue culture there should be a few things written on it. The cell line, the initials of whoever has been maintaining that flask, as well as the last date it was passaged, and then finally a passage number. And that just indicates how many times the cells have had media added on top of them. And although these are immortalized cell lines, after a while, after typically people say like somewhere between 30 and 40 passages, the cells do start to get a little weird. And so it's important to keep track of this passage number, just so you know once you get up to a certain passage number, it's time to throw away those cells and start fresh with a new frozen tube. So for now we are on passage number 14. I'm indexing that up by 1. So these jerkats still have a long time to go. Today is the 28th. And let's see, is this marker any better? Nope. I'll label these plates later. In the meantime, I'm going to grab my jerkat media. So I put this in the water bath a while ago from the fridge to let it warm up. So again, we're going to spray this down with ethanol before we bring it in. And be careful because these bottles do get slippery, just like the glass. But we do want to make sure they're sterile. So I'm rubbing the ethanol all around. I don't want to dry off the ethanol immediately because then it can't do its job to sterilize the surfaces. All right, here we go. So that's perfect timing. Our jerkettes have finished spinning down in the centrifuge. And you should be able to observe a pellet at the bottom of this tube, and those are our cells. So what I'm gonna do now is I'm gonna grab a 5mm pipette to resuspend this pellet in. And out of all the pipettes, the 5mm tends to be the best. And that's just because of the bore size of the tip down here. It's the most narrow, and so when you shoot liquid out of this tip, it creates the most turbulence at the bottom, and that is actually what resuspends the cells. So if you ever need to resuspend a pellet, you should do that using a 5mm pipette, unless your cells are like it's really super sensitive to any sort of like shear force. All right, so the first thing I'm going to do is use the vacuum line to aspirate up the old media. So these are these disposable glass pipettes that we autoclave and keep in the hood. I turned on the vacuum. I'm going to be careful now to aspirate the media without sucking up the pellet. And so if you're worried about sucking up the pellet, the best thing to do is actually just leave a little bit of media in the bottom. It's not the end of the world to have some old media in there. Now I'm going to resuspend my pellet in 5 mils of fresh media. I'm gonna pipette up and down something like 20 times. All right. Now hopefully you should see that this looks pretty cloudy, so we know there are a ton of cells in there. Now what I'm gonna do is grab my P20 as well as one of these little 0.5 mil Eppendorf tubes. And I'm just gonna grab a 10 microliter aliquot of my cells for counting. Make sure that's closed. All right. So lucky for us, we have one of these countesses in the lab. And so this automates cell counting for us. The way we use these is we have these slides, these countess slides, that we will put our cells in after we've mixed them in a 1-to-1 ratio with trypan blue. So trypan blue is a dye that is able to get inside dead cells. And so what trypan blue is going to be able to tell us is our ratio of live to dead cells in this tube. So I grabbed 10 microliters of trypan blue. I'm mixing this up and down 5 to 10 times. And then I'm going to load both sides of my slide, count them in the countess, and then take the average of those counts. Okay, yeah, and you'll see that there's just some nice capillary action that draws our media into like a nice layer on this slide. The countess should autofocus. Here you can see all of our cells. Hopefully they'll pop into focus in a second. Great. And now I'm just gonna simply hit capture once I see that it's focused, and I will record that count. 8.6 million. And you don't always have to measure twice. You could just measure once if you're counting a lot of samples, but since I just have the one, I'll use both sides of the slide here. This side is 9.2 million. So you can see that the count is a little bit different. You also notice that I'm recording the number of live cells, not the number of total cells. And our cells look very healthy. They're above 90% viability, so that's great. So now the average of this is 8.9 million cells, and that's in 5 mils of media. So in total, that means I have, oops, I think that's 44.5 million cells, which should be more than enough. Let's see, Damon stole my sticky notes, so I'm going to grab those notes. I needed 26 million total, and I'm plating 52 wells, 27 for mat. So now I just need to do a quick calculation to figure out how to actually dilute my cells down to 1 million per mil, because that's my desired concentration. So luckily this is pretty easy for us. We have right now 8.9 million cells in 5 mils, giving us 44.5 million cells. So to get to a concentration of 1 million per mil, all I need to do is add about 40 mils of media into that tube. Grab one of these 50 ml pipettes. So what I'm going to do now is add 40 mils of media. Then I'm going to start to plate my cells using a much smaller volume pipette to get more accuracy, so that I can more accurately add half a mil of these cells into each well, which is what I want in the end. So I'm mixing up and down for good measure. I'll mix again later though, since that is such a wide pipette, so it doesn't really give good mixing. Okay. So in fact, let's just use one of these 5 mil pipettes to do this. I know it'll be a little slower, but I like to be accurate here. I'm going to change the speed of this pipette min down to medium, again to give me a little more accuracy. Okay, so now I'm gonna mix starting from the bottom. And I know I was getting dangerously close to the top of this pipette here, so just watch out for that. You don't want to soak your pipette min, or else then everything will get contaminated, and or your pipette min will just stop working. Okay, so let's get 6 mils of culture here, and that'll give us 2 rows. Alright, so let's speed through this part. Okay, so now our jerkettes are plated, ready to take virus, and I am going to now take the rest of the jerkettes, passage them into the new flask, which is this guy. Remember, this is at a concentration of 1 million per mil. Now that's pretty concentrated because the next time I'm gonna passage this flask is on Monday, which is in 4 days. So I can't leave the cells at that high of a concentration over that amount of time, so I need to dilute this further. So in fact, I'm gonna dilute the cells down to 250K per mil concentration, which is a 1/4 dilution. So what I'll do here is I'll add 15 mils of media and grab 5 mils of my cells from this tube. I hit the wrong button there, squirted a little in before I aspirated up. So I'm just grabbed a little extra for good measure there. This part does not have to be exact, luckily. But there you go, science in action. Sometimes we make mistakes, but it's okay. Okay, so yeah, that's how easy passaging suspension cells are. So these cells can actually go back in the incubator now. And just in case Matt needs these cells, I'm gonna very quickly label them. We'll just leave them in here, which it is. Just in case. Now, I'm going to grab my LX293Ts, which I transfected on Tuesday. So after 48 hours, they are now ready for me to harvest the virus off of them. So there's a couple things that you want to be looking for here. One is you can actually look under the microscope to see how many of your cells are dead. It's not a very like quantitative measure of viral titer, but let's just say it's a bad sign if all of your cells on the plate are still alive. And that's the reason behind that is because when you're producing lentivirus, the cells will actually lyse and explode, releasing all of the lentivirus into solution. So all of the lentivirus is actually just now living in the media in these wells. And so what we need to do is filter out all of the dead cells and other debris that are in suspension in this plate to filter out the big chunks of cells and other things, leaving the lentivirus behind. Because these lentivirus are very, very tiny, they will pass through a filter while the other cell debris won't. So you can use these filters that we have here, these 0.45 micron filter units. Some people will actually just collect the viral supernatant from these plates and spin it down at high speeds. Well, I guess not high speeds. I'm actually not sure what speed you spin it down at. Pellet the debris and then just take the supernatant off of that. I've just always done it this way, and I think it's a little bit cleaner, but if you want to save money on resources, these filters are quite expensive. That can definitely be one way that you can save. So before I get started transducing my cells, I'm going to quickly label this plate. Looks like my marker is dead, so I'm going to grab a new marker. I'm going to label the plate with what virus is going into each well, and then I'm going to use a syringe and one of those filters that I just showed you to harvest the supernatant, and then actually just put all of the supernatant directly on top of each of these wells. Now, if you're harvesting more than one well of virus, then it probably makes more sense for you to filter into some sort of receiving tube and then aliquot from that tube into this plate. But since I'm doing 1-to-1, one well of virus from this 6-well plate into one well of my 24-well plate, I'm actually not going to harvest into a tube. I'm going to take the supernatant and filter it directly on top of the cells here. All right, so we're back. All of my wells are labeled. And earlier what I did was I gathered a bunch of stuff in this box here. And this includes these syringes, which we'll use to harvest the viral supernatant, as well as a bunch of these filters, which we will then filter the supernatant directly onto my jerkat cells. So I have a couple tips here for you guys. One is to do these one at a time. Open both pieces of packaging before you begin. I'm going to actually try to clean up my pit a little bit first, because it's always a good idea to give yourself as much space as possible when doing this. So I'm going to throw this flask away and put away this media. Giving me a little bit more space in the hood. Alright, so I've popped open both packages. Now again, this is live lentivirus, so be very careful when you're harvesting. Wear proper PPE, have bleach on standby, and also be ready to soak up any spills with a Kim wipe soaked in bleach. Okay, so I tilt the plate. I gently lift up on the syringe to harvest all of the supernatant. You'll see there's a little bit under 1.5 mils of volume here. I'm gonna screw on this filter and then very simply dispense all of that filtered supernatant onto the proper well of my jerkats. Then I'm going to take this syringe, fill it with bleach, and then I'm going to let that sit for somewhere between 15 to 30 minutes. That's it. Rinse and repeat. This part is very simple. It just takes some time to filter every single one of these wells of virus. One tip that I didn't mention, that I actually didn't do there, you might have seen me struggling to get this syringe unstuck at the beginning, is actually when there's no liquid at the bottom, just to move this up and down a couple times, and then you'll see now that I have a much easier time drawing up the supernatant. The other thing that you might be noticing is my grip on the plate. You want to keep the plate as stable as possible as you're futzing around with the syringe and drawing it up. The last thing you want to do is jostle your 6-well plate really hard and either spill lentivirus everywhere or mix up lentivirus between wells. Yeah, that would be no bueno. Alright, we're back. So I've completed transferring all of the viral supernatant on top of my jerkats. And before I put my jerkats away, I just wanted to very quickly mention a few things that some people will do when doing these viral transduction protocols to improve their transduction of their cells if you have a particularly hard cell type to transduce. And those can include, one, concentrating the viral supernatant to get a much more of this viral supernatant onto your cells. And that can help if you're having trouble with your viral titers, which can happen with larger plasmids. And so there, maybe I'll make a separate video on how to concentrate virus. essentially what you do is you combine a large volume of viral supernatant with a concentration reagent, spin that down, and then resuspend in a much smaller volume. Other things you can do are spinduction. So you can now actually take this plate, now that the viral supernatant is on top of the cells, spin it down at very high RPM, something like 1500 Gs, to encourage the virus to settle more towards the bottom of the plate. And that can increase local concentrations of virus around your cells. Another thing that some people will do is like coat their plate with polybrine, which is a reagent that can again improve viral uptake for your cells. For me, with these Durcats, I usually get pretty good transduction efficiency, so I do none of these. In fact, I'm just going to leave the virus on my plate for 24 hours, and I'm going to come back tomorrow and remove the virus and replace it with fresh media. So for now, these cells are going to go in the incubator, and I will see them tomorrow. Now, the last thing that I'm going to do before I sign off for today is just talk a little bit more about the bleaching. So these syringes have now been sitting in bleach for about 15 minutes or so. And so what I can do now is transfer whatever bleach that was in here into these plates. And in doing so, I am going to now clean up whatever leftover virus might be floating around in these wells. And then I'm going to let the bleach sit in these plates for, again, 15 to 30 minutes before aspirating. At which point you can very simply just use one of these glass pipettes and your vacuum line to aspirate up this bleach into your waste flask. And these syringes, depending on the protocol of your lab, should just be able to go into the regular biohazard and not the sharps. But again, this might vary from place to place, so don't quote me on that for your exact institution or setup. So that's about it. I'm gonna finish cleaning up here, but otherwise that concludes how to do a viral transduction. Thanks. Hey everyone, it's Andrew, and I'm back now for a quick update on the transduction. So it's been about 24 hours since I put virus on our cells, and so today what we're going to do is just very simply take off the viral supernatant and replace that with fresh media. And the reason we do that is several fold. So one is that the lentivirus can be toxic to the cells if you give too much to the cells. So just many integrations into the genome of our cells can screw them up. And then the other is just that the viral supernatant that we fed to our cells yesterday is very nutrient poor, since our HEC293Ts were previously living in that media for about 72 hours. So our cells also need to feed. So we're going to put them in fresh media today and yeah, leave them over the weekend. And so even though it's been 24 hours since we harvested our lentivirus, there may still be some live viral particles in the media, although unlikely. And so we still need to prepare our bleach solution to dump the media into, just as a precaution. So just as before, I'm preparing like a 10 to 20% solution of bleach. Going to sterilize this and bring this into the hood. So yesterday we transduced Jurkat cells, which are a type of suspension cell. If you were transducing adherent cells, this would be a little bit easier. But what we're going to do today is essentially take off a little bit of media off the top of each of these wells, spin the plate down, and then take off the rest of the media and replace with fresh RPMI. Now, if your cells were adherent, they'd be stuck to the bottom of the plate, so it'd be very easy for me to just simply aspirate everything off into the bleach and then replace with fresh media. Here, we want to be careful when we're aspirating the media here that we're not taking all of our cells with it. Now, over 24 hours, the cells will have, through gravity, mostly just settled to the bottom of the plate. But you'll notice that there's pretty high volume in each of these wells, maybe like 2 mils worth approximately in each of the wells. So it's a little dangerous for me to spin the plate right now, because when the rotor and the bucket spins, the plates kind of tilt this way or this way, and we don't want media from one well spilling into another. So before I do that spin to guarantee that all my suspension cells are actually on the bottom of the plate, I'm going to take off about 1 mil off the top of each of these wells, and then spin. Oops. Let's see, these tips aren't right. Now, when you're doing this, just remember to pipette very slowly, otherwise you'll disturb the cells at the bottom. And then the other thing you'll notice I'm doing is that before I release my pipette into the sharps container, I'm pipetting up and down in the bleach about 5 to 10 times to kill any viral particles that might be stuck to the side of my pipette. Okay, so I'm going to finish the rest of this plate, and then I'll be back to show you how to spin the plate down. Alright, I'm back. So I've finished taking 1 mil of media off of my plate. So now what I'm going to do is spin whatever's left in the plate down. So because there is the chance that there is still live lentivirus in this plate, what we need to do is make sure we're using secondary containment when we put this plate in the centrifuge to make our lives easier if there are any sort of spills, and that there have definitely been spills that have occurred with me before. So you'll see that I switched out the buckets, and these buckets have these nice little lids that come with them. So this is my balance plate. Pop that lid on, and then similarly with my plate that has the cells, I'm going to put it on top of this handy dandy little homemade plate remover thing, which is just happens to be a pipette tip lid with some tape on the sides of it. Gently place that in the bucket, and similarly pop this lid on. Make sure it clicks on both sides. So here you'll see what I was talking about earlier with when this centrifuge gets up to speed, the buckets are going to spin out like this. And so if our plate was full of media as before, there would have been a high likelihood that media would have spilled between wells. So if you ever want to be like super sure that there won't be any sort of cross-contamination, then the best thing to do would actually be to transfer your cells from the plate into like falcon tubes or something, but that takes a lot of time and wastes a lot of plastic. So I found that this method works good enough. If you go through this additional step of taking off a mil of media before you spin, then I haven't had any issues with any sort of cross-contamination then. All right, so I'll be back in 4 minutes when this centrifuge is done spinning. All right, so our spin cycle is done. So we are going to now rescue our plate from the centrifuge. Again, be very gentle here with taking this lid off. Now you'll be able to tell if any liquid was spilled between wells, actually just by looking at the lid, since there is a little bit of like condensation there. If any of that condensation looks wet, or if you just see any liquid stuck in between wells, then you'll know that, well, you better watch out for that. But luckily I don't see any of that happening here for our plate, so we are good. So now the final thing to do is to quite simply remove whatever media is left and replace that with fresh media. So here, hopefully now you'll very clearly see a little pellet at the bottom of your plate, and it's not completely necessary to remove all of the remaining media in here. Definitely keep your eye on that pellet as you are aspirating, because the last thing you want to do is suck up all of the cells that you worked so hard to transduce. So what I'm going to do is very slowly aspirate until I see that pellet start to move, at which point I'm stopping. So I'll typically do like a row or two at a time of my 24 well plate, and then add 2 mils of fresh media on top of my cells. And the amount of media that you want to add onto your cells, I think, is just dependent on the passage conditions and just experience that you've had with your cell line. I found that with jerkats, if you add 2 mils of media on top of them on Friday, they'll survive until Monday. The media will look a little yellow, but the cell viability should be fine if you plan to passage the cells or run whatever assay you want with them on Monday, which is about 72 hours post-virus removal. So the cells will already be at like peak expression by that point. So here I'm just going to show you, I finished the first row, so I'm going to adjust the speed of this pipetter down, add 2 mils of fresh RPMI on top of each of these wells. Here, just be careful to not touch the tip of your pipette into any one of these wells, and then you can use the same pipette for multiple wells. Great. Okay, so now just rinse and repeat. And that's it. That concludes how to do a transduction, and like I said, about 48 to 72 hours post-virus removal, you can feel free to run whatever assays you want on the cells. So there you have it. Thanks for watching.